Hey guys, what's going on? Mike the Caveman Q here again, Paleo Problem Island, MikeTheCaveman.com. Another edition of Monday Nights with Mike, live stream Q&A. Ask me anything, health related ideally, but you know the deal. So obviously it's open Q&A, you can ask me whatever you want, but as we've been doing for the last couple months actually, what we're going to be doing is looking at studies to help us out and break it down, help you translate some of that out to you guys. But again, if you have any questions either directly about the study itself or about anything you want, let's get it going. So today we're going to be looking at a paper entitled Bad Luck Mutations. DNA mutations are not the whole answer to understanding the cancer risk. So yeah, this is Trosco and Giuseppe Caruba. This is from last year around April, June of 2017. So, excuse me. All right, Timmy, what's up? Now, bad luck mutations are DNA mutations, which may not be the entire answer, but it has been proposed that many human cancers are generated by intrinsic mechanisms that produce bad luck mutations by the proliferation of organ-specific adult stem cells. So basically, one cell screws up, and then in the process, based on a mistake, and from there, that particular cell causes everything else to get messed up. There have been serious challenges to interpretation, including multiple extrinsic factors thought to be correlated with mutations found in cancers associated with these exposures. So perhaps that's not correct. While support for both interpretations provides some validity, both interpretations ignore several concepts of the multi-stage, multi-mechanism process of carcinogenesis. Namely, that one, mutations can be generated by both errors of DNA repair and errors of DNA replication during the initiation process of carcinogenesis. Basically, when you have cells, if they start to malfunction, if they have a mutation, generally your DNA first repairs it, and then when it repeats it, it doesn't, transcribe it wrong. So there's translation and transcription. So it makes sure that it goes and proofreads that DNA and make sure that it was done right. So number one, that's the first step, that it skipped over those processes, that your editors didn't catch the mistake in your work. From there, we have two, initiated stem cells must be clonally amplified by non-mutagenic intrinsic or extrinsic epigenetic mechanisms. Basically, the environment or other factors go in and cause it to keep going. So that cell, even after it's met through the proofreader by the editors, it keeps going and is still messing up. Number three, organ-specific stem cells numbers can be modified during in utero development, thereby altering the risk to cancer later in life. Basically, that when you're basically being born, or before you're born, when you're in utero, the number of stem cells you have can be changed by different things. So the more you have that could be messed up, the more likely it is that you're going to end up with cancer. And number four, epigenetic tumor promoters are characterized by species, individual, genetic, gender, developmental state specificities, and threshold to be active, sustained and long-term exposures, and exposures in the absence of antioxidant anti-promoters. So basically, number four, those are the environmental factors. So different things in the environment can promote cancer based on things such as what species you are. We're humans. What are your individual genetic? So what's your DNA say? Gender, because yeah, it's a real thing. Developmental state, so different things, what point of life you're at. Levels of activity, sustained and long-term exposure. So are you acutely exposed to a toxin or is it a long-term thing? And from there, are you exposed to antioxidant things? So antioxidants that will help to turn down that DNA damage. Will, Timmy, Sam, Ozzy, what's up? All right. So because of the inability of some of the stem cells initiating mutations by either errors of DNA repair or errors of DNA replication, a tumor is formed depending on the promotion phase of carcinogenesis. So again, depending on basically the fact is that no matter what you do, at some point, you're going to miss, your DNA is not going to catch a mistake. So if you write often enough, eventually you're going to make a typo and you're not going to notice it. So you get the best spelling, best punctuation, best grammar, and if you don't go back and proofread your work, and even if you do, eventually you're going to mess up at some point. So that's what they're saying. It's inevitable. Doesn't matter what you do, you're going to get cancer at some point. But generally, your DNA can fix that. So what we're saying is that we can't really do much about the initiation phase. You can reduce it a little bit, What's more important is focusing on on promotion phase. So when it goes to the next step. So while it is possible, as I just said, to reduce our frequencies of mutagenic initiated cells, one can never reduce it to zero. Because of the extended period of the promotion phase of carcinogenesis, strategies to reduce the appearances of cancer must involve the interruption of the promotion phase of these initiated cells. So that's the key of this paper, is that can't do much about actually the fact that you're going to have cancer cells. You're going to have typos. But what we need to do is actually prevent that promotion. So let's look into that. Excuse me. So, introduction. Are mutations the drivers of human carcinogenesis? And that's a big debate, whether cancer is a genetic or metabolic disease, right? And obviously, myself, I lean more towards the metabolic end, but more on that in a little bit. So, in the context of understanding the causes of human cancers for the ultimate purpose of prevention and treatment, more and more current paradigms, concepts, and sophisticated technologies are being used to develop the practical means for precision medicine, which, if any of you are interested, I've talked about that before. I actually did a whole doctorate level thesis on that at one point. It wasn't my full thesis, but it was for one of my classes. And precision medicine, I honestly believe, is the future of medicine. Basically, being able to pinpoint things down to your DNA, your environmental exposure, and to your microbiome. So, big stuff. But moving past that for now, in order for this to work, however, for translational clinical trials at the individual level, or for broad public policy to reduce the population burden, it should be safe to say that one should not apply what one does not know. Therefore, it should be prudent to understand as much as possible the basic scientific mechanisms of the complex carcinogenic process. Basically, if you don't know how it works, you can't really make too many decisions based upon that. So it is of utmost importance that we understand exactly how it works. Now, in the context of understanding how exposures to any agent, for example, radiation, chemicals, or microbiological toxins, an organism will manifest a pathologic response. The exposure dose, the time of exposure, the routes of exposure, the individual genetic background, the gender, developmental state, confounding additive and synergistic or antagonistic mixtures of exogenous or endogenous agents must all be considered. So not only are the specific things that you have, that you're exposed to, how long it was, how it got there, what your DNA says, what gender you are, how old you are. Are you young? Are you older? Are you in the middle of your life till your developmental state? Because then what happens from there? There are additive and synergistic or antagonistic, so they can work together. It can be multiplying on top of it, or it can be reducing it. So, and then the endogenous or exogenous agents are internal and external agents. So, in the case of human carcinogenesis, it is assumed up front that cancers are not the result of a one-hit process. Basically, one mistake isn't going to necessarily lead to cancer, right? Now, given the well-accepted and experimentally demonstrated concept of the multi-stage, multi-mechanism process of carcinogenesis, mutagenesis, cytotoxicity, and epigenetic alterations of gene expression, they occur in the stages including initiation, promotion, and progression stages. Hey, Tavlana, what's up? So, the question arises, is or are any of the three basic toxicological mechanisms involved in the pathogenesis of cancer dependent upon the linear no threshold or dynamic threshold or some hermetic phenomena? So basically what they're saying is, does it happen over time, or, as I said, it's not a one hit? So how do we know what progresses the development of cancer? So with the pressures of today, given the global metabolic disease crisis, including cancer, one emerging though aged idea is that mutations are the drivers of human cancer and potentially other diseases. New technological screens to identify several genes when mutated are being used to alert a person that they may be predisposed to one type of cancer or not. With a small percentage of cancers having a major mutated gene that predisposes an organ-specific cancer, and the fact that virtually all cells within a tumor contain mutations, it has unfortunately led to the conclusion that these mutations are indeed the drivers of human carcinogenesis. So basically what we're saying here is that the older model, although technically Warburg probably preceded this model, but whatever, the metabolic, has been around since the 30s, but that kind of got pushed away. And we've been pushed on this whole genetic driving of disease, particularly of cancer. And so what we're saying here is that perhaps, even though there are mutations in all cancer cells, or the vast majority, virtually all, and while we do know that there are certain genes that if you have a high cluster of those genes, you're probably going to have a mutation rather thereof of that gene. You're probably going to be likely have a higher rate of high propensity towards that specific type of cancer. Do we know for sure that's it? Because it's only a small percentage of cancers that actually exhibit that. So to go with a genetic basis as the definitive driver may be a flawed model. But moving forward, with racial, ethnic, and geographical associations of certain kinds of cancer, the emergence of non-genetic, environmental, social, and cultural determinants need to be investigated as to how these factors might influence the role of mutations. That brings up this commentary, the idea that epigenetic mechanisms should be viewed as the rate-limiting step of human carcinogenesis. Basically, what they're saying is that while you have that DNA template we talked about before, right? Your DNA is your architect. It tells you how life should go, so how it should be built, but it doesn't necessarily tell you what kind of small changes the independent contractors, everything else are going to make. So while genetics could be the foundational aspect of cancer, we don't know for sure whether or not it's more important than the metabolic aspects. So in brief, this hypothesis does not negate the role of mutations in hereditary or somatic human cancers, but rather focuses on the epigenetic role of natural hormones, cytokines, and growth factors, and synthetic pollutants, food additives, medications, diet, exercise, stress, chemicals, which can alter the expressions of genes and ultimately play a role in the appearance of a tumor. So again, lifestyle, dietary exposure, environmental exposure, and then just natural things that happen in your life, whether it be hormones, your immune system, and just other growth factors. They can all play a major role on turning on or turning off those genes. So whether or not you have that innate mutation, it may not be the final death knell for the development of cancer. Another problem that must be answered is how to explain certain characteristics of cancers in order to find the most efficacious prevention and treatment strategies. To illustrate, this is with one example, is the observation that human colon cancers arise from the right ascending colon are not arising from the right ascending colon are not treatable. Possibly by understanding the role of stem cells, mutations in the initiation phase of this multi-step multi-mechanism of human carcinogenesis, the normal physiological roles of different regions of the colon, and the roles of the gut microbiome, one might develop testable hypotheses as to see how understanding the roles of mutations and epigenetic mechanisms could influence cancer prevention and treatment. So, again, breaking that down. Excuse me. I'm going to grab some kombucha real quick. What they're saying there is that rather than simply saying, okay, you have these genes that are associated with colon cancer, let's try to figure out why, when you have colon cancers, particularly of the right ascending going up colon, why is it such that we have these mutations, but in the sense of why do we have different aspects of what's physiologically going on with that particular region of the colon that makes it untreatable? What is going on in your gut microbiome? Now, gut microbiomes, what's going on with the bacteria? What kind of changes in your bacteria are important? So basically building that together, putting together a better model. So let's take a look at the role of those bad luck mutations in human cancers. So in humans, there are many different types of cancer as different types of cells in the body, over 200 having diverse risk factors, incidence rates, and geographic distribution. Tomasetti and Vogelstein proposed that the striking variation in lifetime risk of developing different types of cancer is primarily a consequence of a divergent number of non-cancerous stem cell divisions leading to random mutations during DNA replication. Peculiarly, let's try that again. Peculiarly, let's try it one more time in English. Why the hell can't I say peculiarly? Peculiarly, whatever. You know the damn word. The authors insinuate that bad luck is a major determinant over environmental inherited factors in at least 2/3 of tissue cancer risk variation. So basically, what we're saying here is it how you won genetic lotto, or is the environment more important? So is it just a random mutation? Is that the real driver? Firstly, it ought to be emphasized that among the 31 tissue types considered by the authors, the resulting R tumors, or replicative, having low risk score, account for a mere 18 to 20% of all human cancers, while the resulting D tumors, the environmental or inherited, with a high ERS, along with other neoplasms such as breast and prostate, strongly dependent upon lifestyle and environmental factors, but not considered in the study, account for the 55 to 60%. So those that have more lifestyle and environmental factors account for the other, or not the other, but account for roughly 55 to 60% versus the purely genetic ones where we know which gene is jacked up. Those are 18 to 20%. So, secondly, although the supplementary material provided by the authors indicates the source of the data used for the sensitivity analysis, calculations were made largely through arbitrary assumptions based on the variable results in literature. Furthermore, it's unclear why human tissues such as breast and prostate, despite the characteristics of their stem cell populations, have been investigated in a number of studies, have not been included in the analysis. Anyhow, that's a weird way. I don't know if I've ever seen anyhow in a research paper. That's a weird thing. Whatever. Anyhow, as also admitted by the authors, number and doublings of tissue stem cells may well be determined by environmentally dependent epigenetic mechanisms, whose potential impact is impossible to weigh in the study. So again, the authors themselves, a limitation of their work was that don't really know how much the environment's playing a role. As one could expect, there has been substantive criticism to the conclusions drawn by Tomasetti and Vogelstein. A major challenge to the original idea that differences in inherent cellular processes, namely differential proliferation of organ-specific adult stem cells, are the primary reason that some tissues have a higher risk to develop cancer than others, has recently been raised. So basically, might be something other than the simple genetics. In the history of science, where there have been clashes on explanations for the major problem, in this case, the cause of human cancer, each side has often used some strong evidence to support its case. In this recent clash to find the cause of human cancer, given the important policy implications for prevention and treatment of cancer, it seems that too many in the cancer field today have ignored some classic experimental evidence and concepts that could provide some insights to help resolve this recent controversy. So basically what they're saying is that despite the fact that there could be something here, a lot of times people get caught in their own camp, and that's a huge deal. Way too often people get into their echo chamber and that's all they care about. That's it. I'm not going to hear anything from the other side. This is my team. We get too dogmatic, and unfortunately, people lose in that process. So when people talk, we could get a little bit better. Excuse me. Now, Wu et al., while assuming that intrinsic stem cell proliferation rates and extrinsic factors were entirely independent, asked what happened when environmental exposures affect stem cell proliferation rates. This group used epidemiological data showing how migration of people from regions of lower cancer risk to those of higher cancer risk soon developed diseases at rates consistent with their new home. So basically, if you went from somewhere like, say, Japan and Okinawa in particular that have really low cancer rates, and they move them to the United States, odds are those Japanese, despite having historically low risk of cancer, might actually develop a higher level then. Hey, Doreen, Chrissy, Anthony, what's up? All right. So following up on the original paper, in response to the many criticisms to the original paper, Tomasetti and Vogelstein analyzed different categories of cancer causation, hereditary, environmental, and mutations of stem cell replication on a large dataset of international cancer frequencies. While in agreement with the idea that errors of DNA replication of stem cells probably play a major role in human carcinogenesis, their analysis still failed to take into account the multi-stage, multi-mechanism process, especially the non-mutagenic or epigenetic promotion phase of carcinogenesis, and the role of modulating the number of organ-specific stem cells during development. So what they're saying there is that while these people cared very much about the mistakes, the proofreading errors we talked about in the beginning, what they didn't really discuss was that fact that there were occasions where there were not mutations. There were other occasions where it's the environmental, the epigenetic promotion, the phase of carcinogenesis. So after it's already been mutated, what do we do then? And then the role of changing how many specific stem cells, how many adult stem cells during development are around. So you're able to change that number. All right. Presumptive mechanisms in the initiation, promotion, and progression model of the multi-step, multi-mechanism process of carcinogenesis. So, in experimental mice, rat, and rabbit animal models, it has been shown that carcinogenesis is a multi-step, multi-mechanism process. This led to conceive the carcinogenic process as an irreversible step that occurred in a single cell, which is termed the initiation step. After regular, chronic, and threshold levels of exposure to a different class of agents, and in the absence of agents that negate the effect of those agents, such as and in the absence of agents that negate that effect, which is the promotion step, this initiated cell could then become clonally amplified to a pre-malignant lesion, such as a papilloma in the skin, a nodule on the breast, a polyp in the colon, and then in time, one of those promoted initiated cells could accrue additional genetic and or epigenetic changes that in turn could cause it to invade and metastasize to distal sites, or the progression phase. So basically what we're talking about here, and hey Christine, hey Kev, what's up? So what we're talking about here is that in their model, what we've shown over and over and over again, mice, rats, rabbits, that there are multiple steps. There's the initiation step, whereby there's a mutation in a single cell. Then, that occurs generally after a regular chronic exposure of threshold level, so a certain level of insult over time, and in the absence of things that can rebut that, things that can fix the damage that's been done. That's the promotion step. From there, it gets amplified, it gets recreated on multiple occasions, or replicated. How's it recreated? It gets replicated over and over again. In the process, it then turns into either the papilloma, nodule, polyp, as an example, and over time, and it gets even more genetic or epigenetic changes over time that in turn could cause it to invade and metastasize distal sites. It could spread further. So, there has been no universal acceptance of the underlying cellular and molecular mechanisms for each of these completely distinct operational steps of carcinogenesis. However, because the initiation step process seems to be irreversible, it was only logical to assume initiation came about in single cells by agents that damage genomic DNA, leading to mutation. The classic case of ultraviolet light induction of pyrimidine dimers, which were not repaired, led to the production of gene mutations as errors of DNA repair in the skin cells of human hereditary skin cancer-prone xeroderma pigmentosum. So basically what they're saying here is that this particular hypothesis led to the development of what is the generally accepted model for development of skin cancer in genetically susceptible individuals. Excuse me. Mutations in the oncogene of the skin tumors of these individuals appear to be primarily related to the unrepaired UV-induced pyrimidine dimers. Mutations in the genome of the cells that can give rise to initiated cell can be found in another human hereditary cancer-prone syndrome, the Bloom syndrome. In this case, mutations can be produced by errors of DNA replication. Herein lays a major implication of this process of mutation induction, such as that Caucasians, reducing their exposure to UV light from the sun, human beings cannot reduce their risk to zero of getting initiated cell in one organ or another. So basically what they're saying there is that despite the fact that it's going to happen, these damages are going to occur, the problem here, the major implication is that, in Caucasians, the ability for them to reduce that risk to zero, to completely eliminate a risk, can't happen. But moving forward, another series of experimental findings seems to lead scientists to the conclusion that cancer originated in a single cell. However, regardless of how the mutation is formed in a cell to cause it to be initiated, that single cell is still not a metastatic cancer cell. That single initiated cell is normally suppressed by surrounding normal cells by either the secretion of anti-proliferative factors or by direct gap-junctional intercellular communication, until it is exposed to agents such as endogenous hormones, growth factors, cytokines, and so on, endogenous agents such as pollutants, pesticides, drugs, food additives, solid particles, or conditions that stimulate compensatory hyperplasia, such as wound healing or massive cell killing. So, breaking that down. And did they typo that? No, okay. Just want to make sure. So yeah, so what they're saying there is that normally when this first cell gets its mutations, it'll normally get killed. Okay, so your body will go and see it and be like, no. So the normal cells that are around are going to say, hey, that particular cell's crazy. We need to get rid of him. So while that process is normally cut off in the bud, unfortunately, if nobody around it, none of the other cells do anything about it. If other things, endogenous agents, so was hormones in your body, growth factors, inflammatory agents, so cytokines, and going on, or other agents like pollutants from the outside, pesticides, drugs, food additives, if all those things, if some of them come along and say, hey, you probably shouldn't fix that. It's fine. Don't worry about that. Well, or moving forward from there, the compensatory hyperplasia. So basically, your body sees that and says, huh, there's damage here. We should probably fix this. So wound healing or massive cell killing, what ends up happening is, despite the fact that your body would normally take out that one radical cell, it decided, we'll let them keep going. You do what you want to do. So unfortunately, that's where the problem starts. Now, the removal of this clonal suppression of the initiated cell and its subsequent clonal application is the biological process underlying the tumor promotion phase. This promotion process must also prevent or inhibit the apoptotic loss of these stimulated initiated cells. Promoters classified by the manner that stimulates cell proliferation and prevents apoptosis of the initiated cells at the transcriptional translational level and the post-translational levels. So basically what we're saying here is that these promoters, these are things that are going to cause more cells to proliferate, to keep growing and keep replicating rather, and go out into the world. Unfortunately, these agents then, the promoting agents, can change the way your DNA expresses such that the cells at the all levels, the transcriptional, translational, and post-translational levels, they all go and keep going. I don't think that made sense, but hopefully it did. In this respect, convincing evidence is accumulating that many bioactive dietary components may extensively modulate epigenetic mechanisms, leading to the rapid and effective regulation of gene expression and function in response to nutritional changes. So, what they're saying there is that what you eat may actually change how your genes express. That's the short version there. In this respect, the term epigenetic diet has been introduced to indicate the consumption of foods such as soy. Ugh. Yeah, anyway, grapes, cruciferous vegetables, and green tea that affect epigenetic mechanisms to protect protect against cancer and aging. It is noteworthy that it's noteworthy that an unbalanced maternal nutrient intake may severely impact on fetal epigenome early during in vitro development. So, what mom eats may affect baby's problem or baby's risks for later in life. There is also increasing evidence that nutritionally induced epigenetic alteration of the offspring's epigenome may be responsible for the higher susceptibility to cancer development later in life, and that several epigenetic marks can be inherited and reshape developmental and cellular features over generations, a phenomenon referred to as epigenetic inheritance. So, what they're saying there, like we said, is that not only can what mom does nutritionally change how you're going to develop, so how the baby's going to do, but that actually can be passed on over the course of multiple generations. It's crazy. But moreover, promoting agents such as phoryl esters, dichlorodiphenyl trichloroethane, or 2378 tetrachlorodibenzopidoxin, phenobarbital, phthalates, and so on exhibit threshold levels, species, gender, and developmental stage specificities. In addition, the initiated cell must be exposed to these agents in a regular and sustained chronic fashion. Equally as important, these initiated cells must be exposed to these promoting agents and conditions in the absence of anti-promoting agents that can negate the effects of the promoters. Examples of this class of anti-promoters can be resveratrol, green tea components, and metformin. So what we're saying here is that you need promoting agents, so different chemicals that can promote development and progression of the cancer, but they also need to be exposed chronically over time in the absence of anti-promoting agents. So by not including Things like the green tea components, things like the resveratrol, that's where the red wine comes in. Other compounds, so the anti-promoters can basically prevent the further promotion. That's why they're the anti-promoters. Excuse me. While most humans die before a tumor is detected, all of us have initiated cells in our body in most, if not all, of our organs. Those that develop a tumor in one organ or another before they die had an initiated cell promoted by exposure to some factor, such as specific dietary components, a pollutant, some medication, lifestyle behavior, infection, chronic inflammation, and in addition, not being exposed to anti-promoters. Even all those who develop a cancer can be subjected to these promoting agents and conditions, but, even those who do not develop cancer can be subjected to these promoting agents and conditions, but their initiated cells might be exposed to these agents at below threshold levels, or being exposed simultaneously to the presence of antipromoters. This is to negate genetic or gender factors that can play a role in, I keep saying, now I keep forgetting my knots. This is not to negate genetic or gender factors that can play a role in suppressing the initiated cell from being promoted. So, what we're saying here is that there are environmental factors that can change your risk of cancer because of what's called the allostatic load in general. I've talked about the allostatic load in terms of stress, and it's kind of what we're looking at here. It refers to the threshold at which the amount of stress you've taken in can cause a problem. In this case, it's the threshold at which that cell, so that initiated cell, once it's mutated, it can go on. So it made it past the point where its neighboring cells didn't really peg it as something being a problem. But without something else coming to basically piss it off, whether it be the enzymes, whether it be hormones, whether it be medication, whether it be environmental toxin, whether it be lifestyle factors, whether it be an infection, without something coming to set it off, promoting it to develop into a further cancer, it's probably not going to matter. Or if something doesn't come along to anti-promote, so different compounds, like we said before, then yeah, people may have those tumors, but it never advances to the point where it becomes a problem. Now, again, this is not to negate genetic or gender factors that can play a role in suppressing the initiated cell from being promoted. So, what we're saying here, both factors of your DNA. So, if you don't have a mutation, a basic DNA template mutation, so not a specific mutation that's happened that was a mistake later on, a typo for all intents and purposes. These are what your DNA is already done. So we know, again, that 18-20% we mentioned before of cancers can be directly linked to a specific type of mutation. If you have that mutation, yeah, you probably have a better chance of getting cancer. The other 80% are more likely due to environmental and lifestyle factors. So, genetic is a component. Gender is a major component, and that's something we really need to look into. As we become more accepting and understanding of the transgender issue, no issues with what you want to do with your life, but you have to be aware that there's likely a higher propensity for cancer because we're playing around with hormones that not necessarily a good idea. And what I think is a big problem in that issue is that people can't talk about that. Everybody wants to be as accepting and tolerant as possible. And you know what? I understand that completely. You shouldn't be assholes to people. But that's something that needs to be discussed way too often, and particularly now as it moves towards letting kids transition. That's a point where there needs to be honest conversations about, look, whenever you expose yourself to endogenous and exogenous hormones, or rather increased levels of exogenous hormones, you're gonna throw some things off. You have to do these kind of things very carefully, and you have to really think about it. So gender is absolutely a factor in cancer risk, and it's something that needs to be considered. So just saying. We'll shelf that for now, but just putting it out there. While it is beyond the goal of this commentary, the question of what is that single cell that can be initiated and substantially promoted to become a malignant cell is of utmost importance. The classic stem cell hypothesis against the de-differentiation or the reprogram hypothesis have been offered up as opposing hypotheses of the origin of a single initiated cell. Again, acknowledging the answer to this question is not universally accepted, there is strong evidence that adult organ-specific stem cells exist in the skin, intestine, liver, pancreas, eye, breast, blood, immune system. It is universally recognized that all tumors are composites of heterogeneous cancer and normal cells. And today, most accept that all tumors are sustained by cancer-initiating stem cells or cancer stem cells, while also being surrounded by normal stromal cells, cancer non-stem cells, and other invading cells. These cancer stem cells seem to be more resistant to toxic physical agents such as radiation and toxic chemicals. Excuse me. I'll take another sip real quick. Finally, to the clash between the two opposite views as to the cause of human cancers, mutations do play a role in the initiation of the carcinogenic process. However, with the exception of mutations caused by exposure to UV light that are associated with the mutations in oncogenes of skin cancer or errors of DNA repair, most mutations are probably induced by errors of DNA replication in stem cells that are stimulated to proliferate because of growth, wound healing, massive cell death, or because of exposure to endogenous or exogenous agents, which are the epigenetic promoters, that can cause stimulation of these error of replication or initiated cells. So, recent demonstration of endogenous and exogenous non-mutagenic agents that can stimulate organ-specific stem cells do provide evidence for the suggestion posed by Wu et al. So we'll move over to the picture over here on the side, the cancer iceberg, to really break this down a little bit. What they're saying there is that once you have these initiated cells, which are gonna happen, it happens. There are gonna be typos at some point. Hey, money first, what's up, brother? So at some point, there's gonna be typos. It's just a fact of life. Once that happens, okay, there are going to be errors of DNA replication, right? So once you get exposed to the endogenous, exogenous, non-mutagenic agents, those can stimulate those organ-specific stem cells to basically cause the problem. So you have your primary, your secondary, and tertiary prevention, right? So our primary idea of preventing is through research. From there, we need to integrate that research and clinical instrumentation, which is basically the point at which we can decide, okay, this is what the research says, this is what we see in clinics. And then the clinical instrumentation, basically what we have, where we can decide what's going on. So what is happening, what signs and symptoms can we see to determine that the cancer is basically there. So the cancer iceberg, let's break the figure itself. The whole cancer process can be viewed as an iceberg, where only the minor emerging part can be perceived from either clinical or instrumental viewpoints. Conversely, the bulk underwater part can be appreciated, though partly, by researchers who in turn miss the iceberg tip. Only an integrated perspective can encompass and provide important inferences for cancer prevention, from primary control of risk factors to secondary early diagnosis and tertiary therapy and rehabilitation type of prevention. So what we're saying here is that primary level, that below, is preventative. Stop the risk factors before they happen. Then secondary, that is the early diagnosis, right? So you're going to integrate what's going on and then what happened afterwards. And so how can we prevent it after? And then the therapy and rehabilitation is, okay, we found it. We know we have cancer here. That's that threshold line. At that point, you pass the risk factor part. You now need to work on combating it after the fact. So that's really what we're looking at, is that so far, those levels are separated. There are some that are looking at controlling the risk factors. There's some that are looking at early diagnosis, and there's some that are looking at therapy and treatment. We need to hit all three of those and integrate them together. So, all right. So moving forward, if one assumes for the moment that the organ-specific stem cell is the target for the initiation of the carcinogenic process, then it follows that by increasing or decreasing the organ-specific stem cells, one should alter the risk of cancers. Consequently, the original question posed by Wu et al., what happens when environmental exposures affect stem cell proliferation rates, seems to be supported by both experimental animal and cellular studies showing that endogenous and exogenous epigenetic chemicals can affect organ-specific stem cell behavior. So what happens in life, what you're exposed to, can change it. It has been postulated that the altering organ stem cell numbers, especially during fetal development, might explain the Barker hypothesis. And again, that right there is talking about what happens when mom is doing things. Now, the low frequency of breast cancer in Japanese women during the Second World War, seen in the study of atomic bomb survivors, might have been the result of the unique diet of those women. Caloric restriction, soy product prevalence, green tea, no smoking, vegetables, etc. Since many chemicals have been shown to induce either terminal differentiation or apoptosis of human adult breast stem cells, the exposure to dietary components during pregnancy of the female fetus might have reduced the number of breast stem cells, such that at puberty the breast tissue would be smaller and the number of adult breast stem cells to be targets for the initiation of breast cancer would be limited. So what we're saying here is that in these women, particularly these Japanese women during World War II, as they were survivors of the atomic bomb, you would have expected there to be a tremendous increase. Now, because of their diet and lifestyle factors, there was not a crazy increase. What happened was that there were less stem cells to be initiated, so less pool to start from, less potential for development later on. From there, they also had those non-promoters we talked about before. So with the current change in the Japanese and the diaspora or leaving of Japanese to other countries, they have changed the patterns of breast cancer frequencies compared to Japanese women during World War II. So World War II, bombs dropped on them, lower breast cancer rates than there are today in Japan. And not only in Japan, but particularly in Japanese women who have moved to other countries. It's a crazy idea. Excuse me. The fact that human beings may only partly control DNA mutations by reducing exposure to genotoxic chemicals in the environment or in their diet, but cannot set the zero the risk of developing cancers, rises to the important question as to whether or not it would be more productive to direct public resources to the design and implementation of primary prevention strategies to avoid or restrain cancer promotion rather than to cure cancer. So again, what they're saying there is that those initiated cells, they're going to happen. Can't do anything about it. At some point or another, some cell is going to screw up. You're going to make a typo. And your proofreader is not going to catch it. And from there, at some point, something's going to happen where the cells around it, it's going to do something crazy. And the cells around it don't see that. And they don't stop it from getting any further. So because of that, it starts to get further. Now, when we see that, maybe we should try to prevent it from getting further. Maybe we should be focusing our efforts on that stage, as opposed to focusing on what to do after it's got too big. So that's something we need to look at. In any case, yet researchers and clinicians need to place common efforts in uniting their own knowledge and abilities to create a perspective large enough to enlighten cancer understanding and to provide effectual solutions to major open questions in both prevention and treatment of human malignancies. So, summary. It is proposed that mutations, as a result of both errors of DNA repair and errors of DNA replication, the bad luck variety, do play a critical role in the initiation step of human carcinogenesis in organ-specific adult stem cells. So basically, yes, there are potentials for just, whoops, that one just made a typo. Happened. The process of clonally amplifying the single initiated stem cell, that was a typo there. Single initiated stem cell during the epigenetic tumor promotion process is the rate limiting step of the multi-stage, multi-mechanism process of human carcinogenesis. So, what we're saying here, that process again of preventing that one cell from going out of control and causing haywire for everybody, yeah, that's what we should be looking at. That's the step that changes what happens when that one cell starts to go haywire. We should be focusing on making sure that one cell doesn't get there, because it's not always ever that one cell. It's that one cell now getting exposed to a whole bunch of other bad things. Now, this tumor promotion process depends on exposure to threshold levels, so enough of the promoting agent, in a regular, chronic, sustained fashion and in the absence of anti-tumor promoters. So, what we're saying here, lots of environmental exposure, lots of things that are causing problems. Hey, Dino. Hey, Jason. What's up, guys? So, lots of these promoters, so these things that are going to cause the proliferation and further development of this cancer, you get those over time, regularly, and sustained. Once to hit that threshold level, you get a whole bunch of it over and over and over and over again. And you don't take in factors that can prevent that, the anti-promoters, then yeah, it's going to lead to that promotion process. Now, since this tumor promotion process takes a long time, it is the step by which efficacious intervention for prevention can take place, since it will never be possible to prevent the initiation step to zero level risk. So, because we can't really do much about what your genes are, we can't really do much about those bad luck mutations. They're gonna happen over and over and over again. Can't really do much about the fact that your cells will be initiated at some point. What we can do is focus on including extra, we can do is focus on taking in extra levels of the anti-promoters and taking in less or minimizing our exposure environmentally and dietarily and lifestyle wise to the promoters, right? Now, every time a stem cell is forced to proliferate, especially during development, there can always be a finite chance for an error of DNA replication or a bad luck mutation. This initiation step in the adult stem cell probably stops the terminal differentiation of the stem cell, allowing it to accrue more mutations, both gene or chromosomal, and epigenetic alterations during the promotion stage take place during its evolution to the progression of invasive metastatic step. So basically what we're saying here is that more stem cells in the body means more likelihood of a bad luck mutation. So that typo. Hey, Nick, what's up? So there's more likelihood of that typo. From there, the more it's there, the more time it has to basically build up more damages, or rather more mutations, so gene and chromosomal damage or epigenetic alterations, the more exposure it has to those endogenous and exogenous chemicals, the more time it has to be radicalized, basically. All right. And from there, it can go and invade and cause much bigger problems down the line. So, while strong, unequivocal evidence does not exist showing a nonlinear basis of mutagenesis, it seems that both protective mechanisms preventing DNA damage and known DNA repair mechanisms would suggest the initiation phase must have some threshold. On the other hand, the promotion phase, the rate-limiting step of carcinogenesis, clearly exhibits a threshold dynamic. So, what we're saying here is that it's unclear based on the current research that we know. We don't know for sure, and it's quite possible that it is a linear model, meaning that there is a step-by-step progression that will always lead to that mutagenesis. We don't know for sure, but there's strong unequivocal evidence that does not exist. So basically there isn't strong evidence showing that it's not nonlinear. It's quite possible that it is linear, that it goes bam, 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 bam. But it does seem that after it's been done, after it's been initiated, there is a threshold. So the less exposure you have, the more you reduce that exposure, the better off you're gonna be in the long term in terms of preventing it from becoming a big problem and developing fully into that cancer. So big idea, big takeaway, more anti-promoters, less promoters, focus on your lifestyle factors, and be ready to rock. So, yeah, looking a little slow right now at this point. So we made it to the end of this particular paper. As you know, I'm going to pop it onto YouTube at some point after this, and I'll drop the link for that in the description over on YouTube, especially if you're watching this on the replay. It'll be down in the description. But from there, guys, Fire Cream, if you haven't seen, should be coming out any day now, as I've been threatening for a long time, but you know the deal. We've been calling out for reps, so if you are interested, shoot us over a message over on Fire Cream or shoot me a message personally, and I'll get you in contact with my partner, and maybe we'll get you on the Cream team. Other than that, guys, you know the deal. Shoot me messages, shoot me emails, and ideally pop on for these Monday nights. Yeah, I'll see you next week.